Hello everyone, my name is Hayden Fredericks and these are my colleagues.
I'm Kate Platte.
I'm Devon.
And we are a part of the chemistry department working under Dr. Harris, and today we will be discussing our research on the synthesis of antioxidant modified P188.
Okay, so to get started, we're going to talk about why this is important. So in 2011, the cost of cancer approached $173 billion, and that was the estimated approach by 2020. And then 1.5 million emergency department visits were spent on the top 10 complications from cancer treatments, and the total cost was over $38 billion. And now this is not including non-emergent visits, which could be up to $7 billion additionally. So our goal is to help develop a drug that will help mitigate the side effects caused by cancer radiation for patients. Okay, so what is P188? P188 is a poloxamer. It is a non-ionic tri-block. So right here is hydrophobic, and this region here is hydrophilic. This region is also often referred to as PPO, and this would be referred to as PEO. Okay, so the applications of P188. So P188 right now shows that it helps repair cells after radiation has damaged them. So the FDA has approved P188 for certain pharmaceutical uses, such as mouthwash, even laxatives, toothpaste, and other industrial and pharmaceutical problems. And so this is important because it will help P188 get into clinical trials faster. So poloxamers that are above or below a certain molecular weight will not work. If it's too small, then it'll be too hydrophobic. And if it's too heavy, it'll be too hydrophilic. So P188 falls right into the sweet spot for that. Okay, so this hydrophobic region right here, it will insert into the cell after it has been damaged. These two hydroxyl groups here and here, they can be exchanged for other groups, which is what our team's working on right now. As many of you know, oxygen is electronegative. So that means when it's in an organic environment, it is able to create hydrogen bonds and helps it be soluble and very stable in that environment. Okay, so how P188 works in the body. So when a cell is damaged, the surface pressure drops, and P188, because it is hydrophobic, it is able to insert itself into the lipid monolayer. So then we are working on putting things on the end, functional groups on the end, which we'll talk about later. But when it is inserted, it will help the cell reseal itself, and then once the cell does reseal itself, it is able to be squeezed out. Right here, this is the part that is stuck into here. Okay, so an important thing about this is that although we're only talking about the lipid monolayer, we're not sure if it works for the lipid bilayer, but we're hoping that it does cross over into that as well.
So our proposal for this project is to attach antioxidants onto the head groups of P188, which is our backbone molecule. So I'm Hayden Fredericks, and I will be doing ester linkages with an esterification reaction. And then my colleague Devon has been doing ether linkages to attach these antioxidants onto the ends of P188. And this will just help us by using these two different types of reactions. It will help us to expand our versatility of potential antioxidants that we can attach onto P188.
All right, so in researching the uses of P188, we came across several studies that utilized P188 alongside of targeted antioxidants. So the use of the antioxidants is to react with free radicals within the body to neutralize those free radicals. Otherwise, those free radicals would interact with the cell membrane, which would result in lipid peroxidation, which means that the cell membrane would be damaged. So the reason why we have a variety of these antioxidants is because each one of them reacts differently with the free radicals. And so when these antioxidants react with the free radicals, they produce metabolites, which are non-toxic to the human body, which is useful. So we are attempting to make molecules of P188 with each of these antioxidants attached to the ends in order to increase our chances for the desired functionalization of our molecule. So this is my most recent synthesis with the ether linkage with ascorbic acid. Initially, I replaced the primary alcohol with a bromine, and then I protected the other alcohols with silyl groups, and this was because in previous reactions we realized that the alcohols were very reactive with the solution that they were in, and it created an intractable mix of products. So, okay, so in here I have the second part of my reaction, which is the deprotonation of ethylene glycol here. And we use this ethylene glycol as a model substrate for P188 because it has a similar structure to P188, and it is both cheaper and easier to analyze in NMR. So when I deprotonate the ethylene glycol and react it with my molecule from the other slide, I will hopefully get this molecule, which we are still waiting on testing to see what the results are.
And so with my synthesis, I targeted the antioxidant lipoic acid as shown here, and I was attaching that to P188 as shown here. And then in order to run this reaction, I used DMAP as a nucleophilic catalyst, I used DCC as my coupling reagent, and I used THF as my solvent. And then also you can see that I used a very high equivalency of lipoic acid in terms of P188, which is a 15-to-1 equivalency. And this is because we really wanted to push the lipoic acid onto both ends of this P188 molecule in order to get optimal functionalization of this molecule. And then in this reaction, I ran it at room temperature for 40 hours in the dark. And this reaction ended up producing this desired product with P188 as our central backbone and lipoic acid attachments on both ends. We had a 53% yield and greater than 95% functionalization of P188 with lipoic acid attachments. So this is the NMR data. This is the desired product that was created, and as you can see, we had lipoic acid attachments on both ends of our P188 molecule. So I used NMR to analyze this product, and specifically you can see these purple assignments are the protons that correspond to lipoic acid, whereas the red protons correspond to our large lipoic acid, our large P188 backbone. And so in order to do proton NMR, what we did was set this peak specifically here for the protons on carbon H, as shown here and here. We set that integration to 4 because if you look at our desired product, this is a carbon, it should have 2 attachments or 2 protons attached to it. And this carbon should also have 2 protons attached to it, meaning that it should have 4 protons attached to that. So with that integration, that helped us in order to analyze the other peaks in this NMR analysis. And so then what we ended up using is this peak, which is our methyl group. And this group specifically should have 3 protons coming off of it. 3 times 27 should mean that there are 81 protons shown in this NMR. And if you look at the peak that corresponds, that shows an integration of 81.02. So because we know that NMR data has a 5% error, we are confident that we can conclude we did get our desired product with a 95% functionalization of lipoic acid on both ends of P188. So some future directions for this project. The first thing is attaching other antioxidants. So the other antioxidants that you guys saw earlier in this presentation, specifically phenosin acid, DOPAC, and FCA will be our next potential antioxidants for attachment. And then we also have collaborators at Furman University who are working in Dr. Tim Hank's lab, and him and his team are specifically working on the molecular modeling and characterization of potential antioxidant attachments and how they will interact with P188. And then we also have collaborators at MUSC, which is Dr. Yost and his team, and they are actually working on the biological testing of this molecule. So with the molecule of the lipoic acid attached to both ends of P188, we sent that off to MUSC for some testing. And with preliminary tests, it does suggest that when Dr. Yost and his team used hydrogen peroxide as a free radical stressor to the cell membranes, this actually resulted in tests that showed that the lipoic acid flanked P188 had a higher cell viability in comparison to solely P188 on its own, which is more effective in the control group receiving no treatment. And then our future end goal for this project is to potentially use these desired molecules as therapeutic treatments in conjunction with radiation therapy in order to mitigate any side effects.
All right, so we have a few acknowledgements. We'd like to thank the NSF for their generous donations, Furman University for letting us use their equipment, and Dr. Robert Harris for his expertise and guidance, Dr. Tim Hanks for his guidance as well, Dylan Singhi and Madeline Turnipseed for their molecular modeling work, and Dr. Mike Yost and Kim Sutton at MUSC.